Well, today we will talk about microRNAs during the Natalizumab therapy in multiple sclerosis. The review for the conference, I searched for published reviews and controlled trials on these databases, and I searched from January 2013 to July 2019 with these keywords. Well, when we study microRNAs in experimental autoimmune encephalomyelitis, and when we study this big subject of microRNAs in multiple sclerosis patients, we notice that the number of publications about studies on this topic is increasing year per year. However, what are microRNAs? They are a family of small microRNAs, small RNAs, with on average 21 to 25 nucleotides in length and are non-translated. The fundamental role of these molecules is to regulate the expression of genes either by translational repression or by the cleavage of the target messenger RNA. Here we can see the microRNA gets several reactions from the nucleus to the cytoplasm in its major form, the RISC complex, or the RNA inducing silence complex. And here, the PCR amplification, the reaction that we can use to select, to isolate the microRNAs in vitro. In MS, the expression profile of microRNA is changed in CNS lesions and the immune system. This dysfunction results in considerable effects on gene expression in numerous cell types like B cells, T cells, and onto to mast cells in eliminating diseases. It consequently contributes for worse MS phenotype. The utility of microRNAs within several exosomes as biomarkers of MS is due to the fact that in many inflammatory and eliminating diseases there is a significant increase in circulating exosome concentration. Considering exosomes can cross the blood-brain barrier, circulating exosomes in MS originate and may originate from affected CNS cells. The study of this interaction of messenger RNA and microRNA is very important. This study about this screening in MS patients shows to us that these molecules, the microRNAs in this disease, this study increases exponentially not only to analyze the phenotype of the patients and the pathological involvement of the microRNAs in this disease, but the study of the subject in this disease expand for the evaluation of microRNAs during different therapies in MS, such as Natalizumab or NTZ. This is an important drug in this disease. Natalizumab is a kind of a rescue drug. This is a drug for first-line non-responding MS patients to the first-line drugs. NTZ is a humanized IgG4 monoclonal antibody that blocks the integrin alpha-4 molecule. Its benefit is related to a decrease in homing of lymphocytes to sites of inflammation. Treatment with monthly infusions of this drug reduces disease activity in relapsing remitting MS. The integrin alpha-4 activated by B cells increases the permeability of the blood-brain barrier, which accounts for a greater cell migration and consequent neuroinflammation. There is a reduction of brain inflammation due to the antagonistic effect of this molecule, the integrin alpha-4, during the NTZ therapy. Here we can see the NTZ against the integrin alpha 4 on T cells surface. In addition, B cells are involved in antigen capture and presentation to T cells, cytokine production, antibody secretion, formation of tertiary lymphoid follicles in CNS, delamination, and tissue damage in MS. The central point for this study and our discussion today in this review of B cells in NTZ treatment is the overexpression of integrin alpha 4 on B cells surface. This is very important. So, NTZ is a drug initially produced to reduce T lymphocyte activity via integrin alpha 4 blockade. However, subsequent research confirmed the influence of NTZ therapy on B cells, and therefore NTZ does not improve clinical control of MS only via T cells, but also by blocking the integrin alpha 4 overexpressed in B cells. So, we will talk about today only about B cells, okay? We will focus on the cell. And now we can talk about the major changes in microRNAs during the NTZ therapy in MS. When we study the first paper, we noticed that treating patients with NTZ, it's possible to normalize the expression of two important microRNAs that were upregulated in untreated MS patients. The microRNAs, the clusters 106b25 and the cluster 1792. Another microRNA that was normalized by NTZ was the microRNA 181A. This is a microRNA that was downregulated in MS patients. These two important clusters are related to a decreased number of pre-B cells and mature B cells and an increased rate of apoptosis of B lymphocytes. In this study, mice with upregulated microRNA 1792 in lymphocytes have developed autoimmunity and die prematurely. This microRNA is also normalized by this drug. This microRNA was downregulated in MS patients, and it plays an important role in homeostasis, differentiation, and inactivation of B cells. The second paper, when we study this scientific research, we notice that the authors have showed that the microRNA 642 was upregulated in blood samples of MS patients, and most important, after 6 months of NTZ treatment. This microRNA decreases cell proliferation by blocking the DOHH or the deoxyribosyne hydroxylase pathway, and so this microRNA inhibited the DOHH expression, the B cell proliferation, and enhanced the cytotoxic effect of mimosine. In the third paper, these researchers determined a longitudinal follow-up analysis of RRMS patients during the course of 1 year of continuous NTZ therapy. They have used a stepwise approach with microarray analysis and subsequent quick PCR verification. They described that the microRNAs 18A and 29A were downregulated before NTZ treatment in MS, but after this therapy, 1 year of NTZ, the microRNAs were upregulated in B cells. So, the first one decreases B cell proliferation by blocking the AKT and ERK pathways, and the second one prevents cancer cell transformation by controlling the TCL1 oncogene. This is one important molecule for B cells. Here we can see the relative expression of the microRNA 18A before NTZ and after NTZ therapy. And here the expression of the microRNA 29A before NTZ and after NTZ. Here after and here before NTZ. The others microRNAs were related to other cell types. The researchers also confirm involvement of all identified NTZ-regulated microRNAs in the experimental autoimmune encephalomyelitis model. These microRNAs were associated with disease severity. Here, this paper is very, very important. The researchers, they analyzed the altered microRNA expression, this microRNA pattern in B cells in MS. They compared the expression of more than 1,000 microRNAs in B cells from untreated and NTZ-treated RRMS patients and healthy controls. Two important clusters drew our attention. Yes, they are here again. The clusters 106B25 and the cluster 1792. They were upregulated in untreated MS patients, and after 6 months of NTZ treatment, they were downregulated. The interaction of microRNA and messenger RNA analysis, this analysis revealed that BCR receptor, PI3K pathway, and the PTEN signaling were the key affected pathways. The effect of this drug on immune cells is variable between individuals and over time. This is one important conclusion. The percentage of B cells has varied from 10 to 40% of total lymphocytes in treated MS patients. It might be due to a fluctuation of clinical response to the treatment, in my opinion. This paper has analyzed the circulating microRNA 150 in cerebrospinal fluid as a novel, as a possible candidate biomarker for MS. The researchers evaluated microRNAs in CSF of patients with clinically isolated syndrome, or CIS, patients with RRMS, and inflammatory and non-inflammatory neurological disease controls. Higher levels of the microRNA 150 were observed in patients with CIS who converted to MS compared to non-converters, and in patients initiating NTZ treatment. This is very important. This microRNA controls proliferation and differentiation of B lymphocytes by decreasing the expression of the important pathway CMYB. This is an important molecule to the proliferation and differentiation of B cells. This microRNA 150 is upregulated in untreated MS patients. Remember that. The researchers described that NTZ reduced the microRNA 150 levels in the CSF with a concurrent and one important increased level of this microRNA in plasma. B cells are in CSF and CNS of these patients, so it could contribute to MS in this disease, to this microRNA release on these patients. Here we can see the relative expression and the levels of the microRNA 150 in cerebrospinal fluid before NTZ and after 1 year of NTZ treatment, and the levels of this microRNA before NTZ and after NTZ treatment, 12 months of this drug. These researchers have analyzed the blood microRNA expression pattern as a possible risk marker for NTZ-associated progressive multifocal leukoencephalopathy in MS patients, one important complication in this treatment. They measured the expression of more than 700 microRNAs in blood samples from 19 RRMS patients at 3 time points during NTZ therapy. 2 patients developed PML after more than 2 years of therapy. They observed a differential expression of 3 microRNAs between the PML and non-PML groups after 1 year of treatment. We can see those microRNAs here in those graphical representations. Here we can see the microRNA 320 in PML group and non-PML group, the relative expression. Here the relative expression of the microRNA 320B in PML and non-PML groups, and the expression of the microRNA 629 in PML group and non-PML group. All the representations with statistical significance. This microRNA induces B-cell proliferation and migration besides binding the BAX and BCL2 pathways. These molecules are very important to BCL activity. This microRNA was downregulated in PML patients after NTZ. The BAX actually is a member of the big family of BCL receptors and proteins. These microRNAs contribute to increased blood-brain barrier permeability and neurological disability. They were upregulated in PML patients after NTZ therapy. There was a positive correlation between therapy time and the expression of the microRNA 320. In the last paper, we can see here the mechanism that this microRNA acts in MS. In MS patients, this microRNA is downregulated in B cells. It increases the activity, the expression of this metalloproteinase, the metalloproteinase MMP9, and it enhances the blood-brain barrier permeability. So, our main challenges. The first challenge is to identify the normal levels of the main microRNAs in plasma and CSF, and with this, identify the deregulated levels of each main microRNA in the different phases of this disease. But the most important challenge, in my opinion, is to study the main microRNAs with statistical significance to evaluate these small molecules and prognosis and therapeutic response in these patients. My conclusions are that even now, the role of small microRNAs as biomarkers is still not clear in the diagnosis of this disease. Papers on the subject evaluated groups of few patients, and many clinical studies did not assess microRNA during or after NTZ therapy. Perhaps the evaluation of these molecules and clinical studies with prognosis and therapeutic response could present more significant results. Here are my data to discuss and review possible doubts. Thank you very much for your attention. Here are my group. Thank you. Thank you very much.